Hello. I hope if everyone could hear me. My name is Dr. Ehsan Sharif Begale, and it's an honor to be with you all with this and other TPCF webinars. And it's an honor to have our guest speaker, Dr. Jack Sharkey from Liverpool Preclinical Center. And before we actually start, and before I actually introduce Dr. Jack Sharkey, I'm just, in case if anyone has any questions, please address them to TPCF host, and we will have time at the end for question and answer session. And we are having these TPCF webinars monthly format, and so in the next month, I think we're going to USA, and one of the speakers will be presenting from there. So with that, let's make a start for today's session. It's an honor to have Dr. Jack Sharkey. So he is the Technical Manager and Radiation Protection Supervisor for the Center for Preclinical Imaging at Liverpool University. And his background is in pharmacology and toxicology, and is in particular interested in the safety and efficacy of regenerative medicine. With that, I would like to give the platform to Dr. Sharkey. So the platform's all yours, and it's an honor to have you. Thank you for accepting our invite.
Thank you very much. I hope you can all hear me okay. I'll share my screen.
Yes. Yep, I can hear you loud and clear.
So just before I begin, I'm a little bit worried about this PC. So if I disconnect randomly halfway through, I will just join again on a different computer, but hopefully it won't happen, but just in case.
Yep, we can see your screen as well.
Yeah, I'll just turn my camera off because save bandwidth. Okay, yes, thank you for the introduction. And yeah, welcome all, thanks for inviting me. And I'm going to tell you a little bit about, so our imaging center we have here in the University of Liverpool, which is called the Center for Preclinical Imaging, or the CPI. And so I'll talk about what facilities we have, how we kind of run the facility a little bit, and then I'll show you some case studies about the work we've been doing over the last 5 or so years. Okay, so I don't know how familiar anyone is with Liverpool, but this is the waterfront in Liverpool. So Liverpool sits on the River Mersey. And let me just get a pointer. Yeah, so we have the River Mersey, and this is probably the most famous building or iconic building in Liverpool, the Liver Building. We have these 2 liver birds on top of the Liver Building. And we have 2 huge cathedrals in Liverpool. You can just see the top of one there, and there's a top of one here. And the university is just next to this cathedral. And if you don't know anything else about Liverpool, you've probably heard of the Beatles, and also you've probably heard of Liverpool Football Club. I guess they're the main things that Liverpool City is famous for. I'm not actually from Liverpool myself, but I've lived here for quite a long time now, over 10 years. So yeah, today I'm going to tell you a little bit about myself, I'll start with, and then we'll talk about the CPI, how we're structured and how we run. Then I'll mention what facilities we have in the CPI, and then I'll go on to talk about some case studies of things we've actually achieved and published in the CPI. Okay, so about me. So I graduated from the University of Liverpool back in 2007 with a Bachelor of Science in Pharmacology. I then went to the University of Birmingham and completed a Master's of Science in Toxicology. I then came back to Liverpool University and I got a PhD in the Department of Pharmacology, where I was looking at molecular biomarkers and regulators of susceptibility to drug-induced kidney injury. So after that, I then moved to a different department in the University of Liverpool and took on a postdoc where I was part of the UK Regenerative Medicine platform. So during my postdoc, that's where I really got interested in imaging, and we were trying to develop cell labeling strategies so we could track labeled cells once we'd been administered into animals as potential regenerative therapies. So we not only were trying to develop ways to track cells, but we were also trying to develop imaging methods to examine organ function so we could really see if these regenerative therapies or the cells we were injected had efficacy in our animal models. So we were largely interested in kidney and liver injury, but we have since utilized these methods for different things as well, which I'll talk a bit more about later. And so after this postdoc finished in 2018, I began my position here in the Center for Preclinical Imaging as the technical manager. So the Center for Preclinical Imaging, so we opened in 2015. So we've been going, this is our sixth year now. So we've got our own little area, our own facility within the University of Liverpool. So we're what is known as a satellite facility to the Liverpool University's main animal imaging or animal center. So we're a satellite facility to the animal center. So we have room over here for animals to live. So they come over here, the mice and the rats. They live in the imaging center, and then all the imaging modalities are under one roof here. So we have 3 permanent members of staff. That is Professor Harish Bhoptani. So he's the academic lead. He was actually originally meant to be giving this talk. We have Dr. Mon Maguire, who is the CPI manager, and we have myself, who is the technical manager. So both Harish and Mon are both experts in MRI. I am not an expert in MRI, but I'm kind of the person who's responsible for all the other imaging modalities we have here. So Mon and Harish have got a more physics-based background than me, and whereas my background's more biological and pharmacological and toxicological. So it works quite nicely. So what we have in the CPI, we have 2 main offices. We have a shared office where we have a number of PhD students who are based here. That's where they work. We also have some postdocs that are based here as well. So we have a meeting area where we do our lab meetings and general stuff like that. We have a main lab which contains a number of imaging modalities. We have a surgical theatre for when people need to do surgery on the animals they have here for any models they're making. We have a mouse holding room, a rat holding room. We have a hot room which contains our nuclear imaging instruments. And then we have a hot holding room for animals that have received any nuclear radionuclides. And then we have our MRI room and MRI preparation room. So this is all funded, well, in part by the University of Liverpool itself, but mainly by 2 large grants, one from the Wellcome Trust and one from the MRC, and also more recently the Northwest Cancer Research Trust. So yeah, it wouldn't have been possible for us to have this facility at all without large grants which have been awarded to us. Harish has been involved in them, but also a number of other researchers. So I'll go on to describe what facilities we have. So the main one, and probably one of the most used, is this Bruker 9.4 Tesla MRI. So it's a preclinical MRI. So it can only fit in small animals. It's designed specifically for mice, rats, possibly rabbits, but nothing bigger than a rabbit. So even though it is a preclinical system, the system itself is still huge. It takes up a full room. So yeah, we can do with this anatomical imaging, we can do metabolic imaging, spectroscopy, which is what Harish is an expert in. We can do cardiac functional imaging, which is what Mon Maguire is an expert in. And then we can do all sorts of dynamic and contrast and diffusion imaging. So yeah, these are just a few images. I will be talking about some of this stuff in a bit more detail later on, so I won't go too much in detail at the minute. We have an IVIS imaging system. So that's a Perkin-Elmer system, which is a bioluminescent and fluorescent imaging system. So that can measure bioluminescence and fluorescence separately. What we'd use this for mainly is people will label cells in a lab, so get them to express firefly luciferase is the most common one. And then we can inject cells into animals and then give them the substrate for this enzyme called luciferin. and then we can detect wherever the cells are within the mouse. And similar, you can transduce cells to express a fluorescent protein as well, and we can image that. So we have an ultrasound imaging system too, which is called the S-Sharp Small Animal Imaging System. So that's a Taiwanese company. We mainly use that for looking at cardiac function in the mice. So it's got a nice cardiac package to look at how the heart is affected in mice. But mainly we've been using it to guide injections. So we can kind of do ultrasound guided injections into different organs for our regenerative therapies. So we do some into the left ventricle of the heart, for example, and I'll talk a little bit more about that later on. So we can generate 3D images with that. We can do Doppler images, so that gives us kind of blood flow. We can look at the direction of blood flow in animals. Actually a little bit more about the IVIS. So the IVIS is very sensitive, so you can really detect quite a small number of your labeled cells, for example, but it's got quite a low spatial resolution. So we can generate nice images like this, and we can get an idea of where the cells are. So here we'd say they're likely to be in the brain area, and these areas are probably the kidneys, but we can't say for sure from these images because all we can see is kind of the surface of the animal. We can't really get any idea of depth. So we have also optoacoustic imaging system, which is called the MSOT IVIS Envision system. So yeah, I'll talk about this in detail later on, but it's an optoacoustic imaging system which uses thermoelastic expansion as the principle to generate kind of images of anything that will absorb specifically in the near infrared range, but don't worry, I'll go into that a bit later on. So again, this has really high spatial resolution, and we can really identify specific areas where we can see contrast agents in the mice. So that really allows us to kind of examine individual organ functions. We have a Quantum GX2, which is a really high resolution CT system, so X-ray system, basically. So yeah, that has a resolution of 10 to 300 micrometers, a really high resolution. We can do respiration and cardiac gated imaging on mice in this system as well. And these are our newest systems. So we've only recently, well, it's actually not that recently. So back in 2019, it was now, we acquired these molecule systems. So these are PET, SPECT, and CT systems. So they're very neat. Each system is an individual cube. So the CT would be this one, for example, and the PET would be this one. They have interchangeable beds. So the mouse would go in this bed. It slots into one machine, you do your CT image, and it moves straight to the other machine for its SPECT or PET image, for example. So mice and rats can fit in this, nothing any bigger than that. So the PET has a resolution of 850 micrometers, the SPECT 600 micrometers, and the CT 50 micrometers. So you can do metabolic imaging, functional imaging. So there's lots of options with this, providing you can acquire the radiopharmaceuticals you need. You can do a lot of different things. And that has been one of our drawbacks with these systems, really. So when we acquired the systems, we initially had an agreement with the Royal Liverpool Hospital, which is really close to our imaging centre. And they had agreed to supply us with radiopharmaceuticals. And then once we got the machines, this kind of fell through, and they had lots of issues at the hospital radio pharmacy, and it has since closed down, which meant we had these systems and no way to acquire any radiopharmaceuticals. So it took us a long time to kind of find some suppliers for these and set all this up. And then finally we got all that set up, and then a worldwide pandemic happened. So we kind of got delayed another year. So we've only really very recently started to actually put these systems to use, but I will show you some data we've been generating later on. So it's exciting that we're finally doing that, but it's been quite a long process and quite frustrating at times. Okay, So that was an overview of the systems we've got in the CPI. So I'll now talk about some case studies. So first of all, I'll talk about optoacoustic imaging to study kidney and liver function. And these are two references where we've published this data in recent years. So a little bit about optoacoustic imaging, because you might not be familiar with it. So it relies on the photoacoustic effect. And this effect happens when light is absorbed by a molecule. So we have a laser inside the system that pulses light at various wavelengths. We can set exactly what wavelengths we want to look at. So anywhere between 660 nanometers and 1200 nanometers, we can do 1 nanometer steps if we want. We pulse that laser light onto our target, which usually is a mouse in this case. So molecules within the mouse will absorb this laser light. This causes excitation in the electrons, which move to an excited state. When these electrons relax, they dissipate energy in the form of vibrations. These vibrations generate heat, which causes thermoelastic expansion. This generates a pressure wave, and the pressure wave is then detected by an ultrasound detector effectively. So this is what the system looks like. And in this area here, it's basically a tank full of water, and the water needs to be there to propagate the ultrasound signal, which is generated when that process happens. So when the molecules release this ultrasound wave. So the mice actually have to go in this, which is, it's almost like a cling film barrier. They sit in like a sack inside the water bath, so they don't actually touch the water. They have a really thin barrier between the water, and they're covered in ultrasound gel, so there's no air. So in optoacoustic tomography, air is really a problem because these ultrasound waves do not propagate through air. So you need the water to be basically touching the animal. So yeah, they sit inside this cling film sling, and they have their own anesthetic supply in there, so they stay asleep during the whole time. And it works nicely. So the water's a nice temperature, it's warm to body temperature for them, so they don't get cold. So another thing, before we do this, image the mice like this, we actually have to shave the mice. So we have to completely remove all the fur, because again, the fur will kind of have air inside it, and that'll stop us detecting anything. So we have to remove all the fur or use nude mice. Most people don't use nude mice because they're quite expensive, so we can just shave the mice and remove the fur like that. What we can also do is place a catheter in the tail vein of the mice, and then we can inject any contrast agent. So a little bit more about this before I go on. So there's a couple of endogenous things that are inside the mice which absorb in this near-infrared range. So that's really crucial. Things have to absorb in a near-infrared range for this to work. So that's in between about 600 and 900 nanometers, probably a bit less than that. So hemoglobin, for example, has a specific absorbance spectra in this range, as does deoxyhemoglobin. So you can kind of look at the oxygenated and deoxygenated blood in an organ, for example. That might be something you'd want to do, so to look at organ perfusion. But what we tend to be doing, and what most of done, is injecting a contrast agent that has an absorbance in this range too. So we know the contrast agent, for example, absorbs really strongly at 780 nanometers. We could then unmix for this signal and really look at the kinetics of the clearance of this contrast agent in an organ. So this is what we've done to look at kidney function in mice. So these are what an image looks like. It's a cross section of a mouse. There is a video in a second, so I won't go into too much detail right now. And this is the kind of data we can generate. So time along the x-axis. And these are regions of interest drawn around the kidney cortex in blue and the kidney pelvis in orange. So at this point, this is where we inject the contrast agent into the tail vein of the mice. So you see a really rapid increase in the cortex of the kidney as the dye reaches the kidney. So this dye, I should say, is exclusively cleared by the kidney. So we get a peak in the cortex of the kidney, which we call the Tmax in the cortex. A little bit after this, we see a peak in the renal pelvis as the dye filters from the kidney to the pelvis. And what we've been measuring, or we have measured in the past, is the time delay between these two parameters. And we've been using that as a functional parameter, or a way to look at kidney function, with the idea being in a mouse whose kidneys are working as they should, this time would be very short. But in a mouse that's got kidney disease, this period of time would be extended. So we can kind of use that to judge how severely injured the mouse's kidney is. We've also used another method to look at kidney function by looking at the area under these curves. So we look at the area under the curve in the cortex and also in the pelvis, and we can take a ratio of these two things as another way to look at kidney function. Okay, so this is an example of data we get. So this is a cross section of the mouse. So this is the spine here, and this is one of the kidneys, and this is the other kidney. I know it's maybe not so intuitive, but I'm going to play this video, and you will see the dye appear in the cortex of the kidney first, and then it will appear in the renal pelvis, which is down here a bit later on. So this is just background for now. And then here, so the dye's in the kidney cortex. You can already see it's starting to appear in the renal pelvis. So this is a healthy mouse, so it was very, very quick. This actually isn't the best image, to be honest. We can generate nicer data than this. This is a very early one. And this is really sped up. So this is over half an hour, this scan. We're on minute 23 now. So we image for usually at least 20 minutes. We used to do a lot longer, but we worked out we didn't really need to scan that long. So a little bit about how this study worked, or how a study worked. So we had 16 mice. We gave a group of the mice adriamycin. So adriamycin is a chemotherapeutic agent which is known to cause kidney toxicity, and actually in quite high doses, which we're given here, it also induces liver injury too. So mice received adriamycin, or a small amount of the mice received saline to serve as the controls. Day 1 afterwards, mice were imaged by MSOT to look at the kidney function and also the liver function, but I'll discuss that in a minute. We then gave half of the mice which received adriamycin potentially regenerative therapy. So this is a macrophage-based regenerative therapy. And then 4 days later, we did the same again. We imaged the mice by MSOT to look at the kidney and liver function. We then culled the mice and collected the blood and organs for analysis. So this is some of the kidney data. So this top graph here shows blood urea nitrogen. So this is a biomarker of kidney injury, which would be measured in the clinic to look at kidney injury. So this is after the mice have been culled on day 4, obviously. So in the saline treated mice, obviously it's very low. And in the mice that received the adriamycin, so both of these groups received adriamycin, it was significantly elevated. This first column is the mice that received no regenerative therapy, and these ones received the regenerative therapy. So there isn't really a difference in the blood urea nitrogen. And this is a graph showing histological analyses. So we actually saw intratubular protein casts and fastening a tubal epithelium histologically, and this is just the scores of the histology here. So again, no damage in the saline group, but damage in the ones that received adriamycin. And this is this top graph here is an example of data from the MSOT from a healthy mouse. So again, like before, the cortex in the black and the renal pelvis in gray. So you can see these are quite nice curves, clear normally. And this is a mouse that received adriamycin, so has quite severe kidney damage. So you can see you get the kind of initial peak and clearance, then the dye starts really accumulating in the cortex and not clearing as it should. That's because the kidneys aren't functioning as they should. And again, in the renal pelvis, it initially starts clearing and then it just starts kind of accumulating. And this kind of summarizes the the MSOT data. So I should say this is a change between day 1 and day 4 in these mice. So there's no change in the function of the kidneys in the mice that receive saline, as we'd expect. And in the mice that received adriamycin but not the therapy, the kidneys got a lot worse in terms of function over these few days. But in the ones that received adriamycin and the therapy, they did get worse compared to the saline group, but not as worse. The damage wasn't as severe as it was in the group that received the adriamycin. And if you're interested, it was published a couple of years ago now. And this just shows how the blood urea nitrogen, so the serum biomarker, correlates with the MSOT data. So it correlates pretty strongly and significantly. So the control mice are these ones down here, and then the rest of the ones are up here. So it's pretty promising, really. So what I'm going to talk about next is how we were looking at liver function in these mice. So we initially, well, what we wanted to do, this is a different dye. So here we're using a dye called ICG, or indocyanine green. So that's a clinically used contrast agent too. It's useful because it is exclusively cleared through the hepatobiliary route, so cleared through the liver exclusively. So it's a good dye for us to use to look at liver function. So what we wanted to do initially was actually look at the liver like we were in the kidney and generate some kinetics from the liver, but I'll show you this video. So this is the liver kind of all around here. So you see initially the dye signal starts accumulating in the liver. And then as time goes on, it starts to accumulate in the gallbladder, which is here. And that's because its route of clearance is through the liver, then metabolized, then into the gallbladder, then into the intestines. So this looks nice. It looks like really interesting data. But the problem is it's very, very difficult to identify the gallbladder until you've injected the dye. And that makes, and unless you can reliably image the gallbladder, you can't really make use of this data. So what we actually decided to do instead was look at the clearance of this dye from a blood vessel feeding the legs rather than looking at it from the liver directly. So we're looking at the clearance from the blood vessel as a kind of indirect marker of liver function. So that was the dye appearing and disappearing from the blood vessel very quickly there. So it's not as interesting, it doesn't look as nice, but it's actually a much easier way for us to generate some quantitative data. So the theory is if the liver wasn't working, this dye would be in the blood supply for a lot longer and it would take longer to clear from the blood. So again, this is the same mice as we were looking at before, but here we're looking at ALT, so that's alanine aminotransferase. So that's a circulating biomarker of liver injury. And again, it's elevated in the mice that received adriamycin, but no real difference between the mice that got the therapy or not in ALT. Sorry. And again, histologically, we saw hepatocellular degeneration and necrosis in these mice. And again, nothing in the saline group, but damage in the adriamycin treated mice. So this is an example of a healthy saline treated mouse in this group. So nice quick clearance from the blood. And this is an adriamycin treated mouse. It's not as obvious as in the kidney, but it is delayed, the clearance. You can see that it takes a bit longer to return to baseline. And this is a summary of that data. So again, much like the kidney data, no change in the healthy mice that receive the saline only, but the liver function gets much worse in the mice that receive the adriamycin, and worse, but not as worse in the ones that receive the therapy too. So that's a positive outcome again. And again, this MSOT data correlates quite nicely with the biomarker data. Okay, so something more recently we've been looking at, because Harish, who is the lead of the CPI, his main interest is cancer and looking at tumor perfusion and oxygenation. So I've been trying to use the MSOT to do that. So I've got to admit, I don't have a huge amount of experience with cancer, but so this is actually a huge subcutaneous tumor on this mouse. And what we've been doing is trying to use ICG, so like that dye I was looking at in the liver, to look at perfusion in a tumor. So again, in tumors that are small, so this graph shows small tumor, this is the clearance kinetics of the ICG. In orange is the core of the tumor, and the blue is the whole tumor. So you can see there's no real difference. So very small tumors are perfused quite well, and the very big tumors like this one here, you can see there's no real ICG reaching the core of the tumor. So this is a way for us to kind of examine how tumors are perfused, and that can be useful in kind of predicting the response of a tumor to therapy, because if there's no perfusion, that would suggest that blood is not reaching the cores of the tumor, or not in adequate amounts. So that might mean your therapeutic isn't reaching the tumor as well. So it's important to be able to kind of establish these parameters, but that is kind of work in progress at the minute. We're still trying to work on optimizing this. Okay. Yeah, so sorry, this was a small tumor, this was a large tumor. Okay, so moving on to a different study here. So away from the optoacoustics, and now we'll look at IVIS. So this is bioluminescent imaging and MRI. So we were combining these two modalities here in this study, and this is published fairly recently too. And we were looking at, sorry, we were looking at tracking cellular biodistribution. So in this study, we were looking at MSCs, so that's mesenchymal stromal cells, or mesenchymal stem cells, they're sometimes called, not so much anymore. So MSCs, or macrophages again, expressing luciferase, so we could image them with bioluminescent imaging. And they were also labeled with superparamagnetic iron oxide nanoparticles, so SPIONs for short. So what we did in this study, we administered these MSCs, which have been labeled with SPIONs or this luciferase, via ultrasound guided injection into the left ventricle of the heart. So we administer them into the left ventricle of the heart because we know through our previous studies, if you inject cells, or MSCs in particular, through the tail vein in the mouse, they first of all go straight to the lungs. And because the lungs have lots of tiny capillaries, the cells will often get lodged in the lungs. So all you see when you image the mice is a lot of signal in the lungs and nowhere else, and not maybe in your target organ of interest. So if we inject directly into the left ventricle of the mouse, we actually get these cells to first circulate the rest of the organs before going to the lungs. So we can actually get some of these cells into the organs we're interested in, which is, in our case, generally the kidney or the liver. So this video, I'll play it now. This is an ultrasound image. And you go, this is the heart here. You'll see it moving when I play the video. And this is the needle. So it's actually showing the injection of the cells. So this is quite a precise thing to do, but it is actually very doable and not as difficult as it seems. So you can see the heart beating. And this is the needle going through the mouse's skin. You can see it going into the heart slowly. And then you'll see some contrast in the heart there. And that's as the cells are being injected. You've got to be really slow to inject the cells here because you could cause an embolism if you inject too quickly or too many cells. So you've got to be quite careful. And it was quite amazing to me at the start that you don't end up killing mice doing this. But as long as you do it slowly, you don't get much or any mortality at all, really. So it's quite good. So yeah, after we'd injected these cells, we imaged these mice using bioluminescent imaging and also MR imaging. So bioluminescent imaging to image the luciferase in the cells, and MR imaging to look at the SPIONs in the cells. Okay, so these are MRI images at the top here. And this top row is mice that received adriamycin, so like I was describing before. So they should have injured kidneys. Both of the kidneys should be injured the same. And this bottom row is ischemic reperfusion injury. So this is a surgical model of kidney injury where we clamp the blood supply to one of the kidneys for a certain amount of time and then release it again to let the blood flow back. And this causes kind of damage to the kidneys. So this is day 0. So this is first of all, let's start here. So this is baseline. We don't see any contrast in the kidneys here because we haven't injected anything yet. This is day 0, so immediately after we've injected the cells. And we see, if you can see the black kind of dots in the kidney cortex here, where the red arrows are pointing to. And the same in both of the models. And by day 1 and day 2, we can see that the signal is lost very quickly from the kidneys. And it's very similar in the bioluminescent imaging. So immediately after injection, we kind of see signal all over the mouse. And this is because we've injected in the heart, not because we've injected in the tail vein. So we get nice distribution all over the mouse. And as time goes on, the signal gets weaker and weaker. And that is just because cells are dying. So if the cells are, that's another good thing about labeling cells with luciferase. If the cells are dead, they can no longer express this gene and they'll no longer light up when you inject the substrates. You know, if you're not seeing the signal anymore, it means the cells are dying. So they do die quite rapidly after injection, and that's widely reported. We know that. And these bottom graphs here just quantify some of the MRI data. This is the kidneys in the Adromycin model. So a big increase in contrast on day 1, and then it returns to baseline, and similar in the other model as well. So this is looking at macrophage biodistribution on its own. So again, we see strong contrast in the kidneys immediately after administration, but it disappears quite rapidly. We do see signal in the liver as well here, but I'm not showing that because we're looking at the kidneys in the MRI. And this is some ex vivo MRI imaging. So this is after we've cooled the mice, we've prepared the kidneys to try and generate some really high resolution images of the kidney. So these are the MSCs. So again, immediately after injection, we see nice contrast in the cortex. You see, you don't really see much in the medulla of the kidneys, and it rapidly disappears. And this is the macrophages. Maybe you can appreciate that the macrophages seem to be able to reach the medulla a bit better than the MSCs. And they actually seem to persist a little bit better as well. And this bottom row is the injured kidneys, I should say, sorry. And this top row is the healthy kidney. So I think you can maybe appreciate in the injured kidney as well, these areas of contrast where the spines are actually seem to be persisting a bit longer. And this is just a video scanning through the kidney. So you can see lots of, all the black dots are spions, so cells with the spions in. Yeah, so it generates some quite nice images. So these black in the middle is the blood vessels. Okay, so we also, once we cooled the mice, looked at ex vivo imaging with the IVIS. So this is the only way with the IVIS you can really say for certain that your signal is coming from a specific organ, because you can actually be certain it's coming from the kidney. So these are the kidneys here at the bottom. This is the heart, this is the spleen, this is the lungs. So always you end up with a lot of signal in the lungs just because there's so many tiny capillaries. And this is the liver. So on the left here, this is the MSCs. So we do get signal in the kidneys, the healthy kidney and the injured kidney, but it's very similar. Whereas in the macrophages, we actually see much stronger signal in the injured kidney compared to the healthy kidney. So that seems to suggest that macrophages actually have an ability to home to the injured organ, whereas the MSCs we think are just going there because they're kind of getting lodged there. There's no homing capacity. They're just there as a kind of result of their physiology. It's nothing more complex than that. Whereas the macrophages are actually probably homing to sites of inflammation. So that doesn't mean to say that the MSCs can't have any therapeutic benefit as a regenerative therapy. It just means that maybe they can't migrate specifically to injured areas. Okay, so next I'm going to talk about some of our most recent data. We actually got a study ongoing currently. We're imaging currently. using DMSA in mice. So again, this is looking at using our model of ischemic reperfusion injury, which I just mentioned, and we're using DMSA in these mice. So a little bit about SPECT, if people aren't aware. So SPECT is single positron emitted computed tomography, and it uses effectively radioactive decay to you're detecting the radiation to generate images. So we have a gamma camera that acquires multiple 2D images from many angles around your subject, which are then reconstructed at the end to generate a nice 3D image. So you inspect, you're measuring gamma rays directly, which is not the case of PET, which is slightly different, which I'll tell you about in a second. Yeah, so when radionuclides decay, you get from an unstable nucleus to a stable nucleus, you kind of get a radioactive photon being emitted. And there's lots of compounds that do this. The most common ones are probably technetium-99, indium-111, and curium-67. We're using technetium-99 in this study I'm telling you about. So yeah, we inject the subject with a radio tracer. So in this study, we're using something, well, technetium-99 bound to a compound called DMSA, and DMSA binds specifically to proximal tubules in the kidneys. So we know if the proximal tubules are damaged, we shouldn't be getting binding of this compound. If the proximal tubules are working in a healthy kidney, we should get this compound binding really nicely to the tubules in the kidney. So yeah, we inject this, photons are emitted from that radio tracer in the kidney, in this case, and they pass through a collimator. So this collimator is made of lead or tungsten usually, so that absorbs lots of the radioactive decay, but we let a small amount through, so we know exactly where in our animal they're coming from when they hit a detector, and then we generate images from images from that. So in this study we had, so this was just our first pilot study really, I'm showing you data from. So we had 3 animals that didn't have any surgery. We had 3 animals that had surgery and a 27-minute clamp of 1 kidney, and 3 that had surgery and a 40-minute clamp of 1 kidney. So this 40-minute group should be more severely injured than the 27-minute group. We administered all the mice with this DMSA. So we gave them about 10 megabecquerels through the tail vein. And then we imaged them for half an hour, 4 hours after they've received this DMSA. So these are some examples of the images we're generating. So this is an example from a healthy mouse. So both kidneys are showing pretty evenly. This is one that's had the clamp for 27 minutes. This is the healthy kidney here. This is the injured. And this is one that's 40 minutes. So again, healthy kidney, and you can't even see anything really in the injured kidney here. So this graph summarizes the data. So the gray bars are the control mice, and we're only looking at the function of the right kidney here, so the injured kidney compared to the healthy kidney. So obviously in the control group, it's about 50% across the days. In the 27-minute group, it started off about 40% and kind of decreases and then kind of increases a bit. So it's like the function dropped and then started recovering. Whereas in the 40-minute group, it drops and carries on dropping. So we've probably got an injury so severe in the 40-minute clamp group that the kidneys aren't, or the injured kidney is not going to recover. So that's promising. We're now doing this in a much larger cohort of mice. So hopefully we'll generate some nice data from that. So moving on to some PET studies we've been doing. So again, this is really the first time we've done this kind of study here. So in this study, we were looking at dual MRI and PET imaging. So we have spions, like I mentioned earlier, superparamagnetic iron oxide nanoparticles. But we have a PhD student who is trying to link these spions to radioactive zirconium. So we can detect them with the PET and also with the MRI. So PET imaging stands for positron emission tomography. So again, like SPECT, it uses radioactive decay, but unlike SPECT, PET uses beta positive decay. And that is slightly different because you're not measuring a gamma photon directly. So this decay releases a gamma photon, which is then annihilated when it hits an electron. And when this annihilation happens, you get two 511 kilo electron volt photons emitted at 180 degrees to each other from this annihilation, and you detect both of these photons, hopefully at 180 degrees each other at exactly the same time at two different detectors that are opposite each other. So this means you don't need a collimator, because if they're hitting the counters at exactly the same time at opposite angles to each other, that you know they've come from the same annihilation. So you don't need the collimator to focus the decay. So this means you can use much lower levels of radioactivity. So you're getting much more efficient use of your radio tracer. So this is kind of a schematic of that. So you have a full ring of detectors around the animal in this case, whereas in SPECT, you might have one or a couple of detectors that rotate around the mouse. So in this study, what we did, we labeled the spions with our zirconium, our hot zirconium, in a lab first. We then administered them to a mouse directly. We didn't use any cells in this because it was just a pilot to see if the labeling worked. We then imaged via PET on day 1 or 0, straight after the injection, 1, 5, and 7, and did MRI imaging on 1, 5, and 7 too. So these mice actually only got 1 megabecquerel of zirconium. We wanted it to be much higher, but the chemistry didn't work quite as planned, as you'll see. We imaged for 40 minutes in the PET, and we did T2 star weighted imaging using the MRI. So these are, I'm going to have to go to my pointer, I think. This won't play. Okay, so this is on day 0 using the PET, so immediately after injection into the animal. So you can see the lungs, there's some in the lungs up here. This is the liver. Oh, sorry. Okay, let someone to play. Okay, take my word for it. Sorry. So you're just going to have to look at these images instead, I'm afraid. So we have lungs up here, liver here. And we also have some small signal in the kidneys, which is just here. So the signal in the lungs was not what we'd expect, given we know spions should probably go straight to the liver from previous studies. So this is day 0. On day 1, the signal is kind of gone from the lungs, but what we're seeing is signal starting to accumulate in the joints of the mouse. You can see in the knees and up the spine, but also in the liver. And as time goes on, this signal kind of gets stronger in the spine and in the bones and in the joints of these mice. So that's not what we wanted. And that is most likely because we're injecting a lot of unbound zirconium here, and it's not bound to the spion tightly like we wanted it to. So the signal in the liver will be because we have some of the signal bound to the spion, some of the zirconium bound to the spion, but we're injecting a lot more free spion than we wanted to. So that's not what we wanted, and that's not good. We were hoping we wouldn't see much signal in the bones at all, but it's clear we are. So this data is summarized here. So the black boxes is the liver, this next one is the lungs, then the spleen, and then this next bar is everything else. So this largely reflects the bone signal. So on day 0, mostly the lungs, the liver, the lungs, and the spleen. After that, the signal basically moves away from the lungs and the spleen and into the bones. So the bone signal gets really large. But the liver signal does stay relatively consistent over the days. So that would suggest that even though we are injecting a lot of free zirconium, much of it is still bound to the spion. So that's promising, but we're getting much more in the bones than we'd like, which isn't good. So we're kind of going back to the chemistry now, and the PhD students going back to work out why so much zirconium isn't binding to the spions. So this is the MRI data, so just showing the liver. So this is the baseline, and this is 24 hours afterwards. Yeah, and then day 1, 5, and 7. So very dark, very much, a lot of contrast on day 1, day 5, and then day 7. And this is just the MRI data summarized. So baseline, not a lot of contrast, and then day 1, day 5, and day 7, a lot more contrast. So it's clear the spions are going to the liver just as we expect. It's just, we have an issue that a lot of zirconium is not staying in the liver, or is going to the bones as it's not bound, so that's not good. So very quickly, I'm aware I'm going on a bit here. This is a PET image of FDG. So FDG is probably one of the most commonly used PET agents. So it's basically radio-labeled glucose. So that's really good for looking at examining cancers, because cancers have an extremely high metabolic rate. So cancers will take up this radio-labeled glucose much more strongly than normal tissue. So in this mouse here, it has a big tumor on its leg here, which is shown there. See if I can get this to play. Yeah, there you go. So yeah, the tumor is on the leg of the mouse there. And we do get some signal in the brown fat, but that's quite normal. And the heart as well, because obviously the heart's got a high metabolic rate too. And this is a dynamic study using FDG. So you can see it quickly is taken up and signal starts appearing in the kidneys, and then as it cleared, it starts appearing in the bladder as well. I mean, this isn't particularly useful with FDG. It was just because I'd never really used the PET before, so I was trying out some scans. Let me just... Okay, so, and this is actually a mouse that's got a brain tumor. So you can see the brain tumor in the head of the mouse here. So it's quite nice. As you can see, the kind of regions on the outside of the tumor have a more intense signal than the regions on the inside of the tumor, and that's probably because the tumor has got a necrotic core, because that's quite a common trait of tumors. So this can actually be observed using FDG imaging. So that's a real good a real advantage of PET imaging, that you can kind of get ideas of what's going on inside the tumor itself. Okay, very quickly, final thing, and then I'll finish. So this is just we had a user that has a specimen which is an Egyptian fruit bat, and she's in the anatomy department, so she's really interested in the muscle structure of the bat. So she wanted to see if she could generate some nice CT X-ray data for the bat. So yeah, CT just X-ray basically, so like you'd have in a hospital if you broke a bone basically. I won't go into how it works too much. So yeah, the X-ray camera moves around the mouse in this case, and we can move the camera closer or further away from the mouse to get higher resolution images. And so this is the bat I was just mentioning. So we're scanning from the top of the mouse, the bat, sorry, through its head all the way down through its body. So I'll play it now, hopefully. So the first thing you're going to see is kind of the wings. So this is like the tip of the wings appearing here. Then as we move down, these kind of white lines appearing are like the skin of the wings, which are wrapped around the bat. The bat's dead, by the way. It's a specimen that's been soaked in iodine solution. That's how we get nice soft tissue contrast. So in a second, this is the head. In a second, you'll see its eyes at the top. Yeah, so these are the eyes. Now you'll see the nose, the nasal cavity, the sinuses, which are really interesting patterns. So now we're moving down into the kind of the body of the mouse, the bat, sorry. And I can't say I'm an expert on bat anatomy, but I think what we'll be seeing in a second. So this is the rib cages coming into view here, or the rib cage. This must be the lungs, this black will be air, so the lungs. That's possibly one of the kidneys there. Yeah, and as we're moving down, we're probably past the liver now, or around the liver, and we're going to start seeing the intestines in a second. Yeah, so these are the intestines. Actually, in fact, these are probably the kidneys, sorry. So yeah, that's a really cool image I thought that we generated with the CT. So we do have another user that comes, and she's a musculoskeletal biologist, and she brings fish, frozen fish, to scan, and she can kind of generate nice kind of data of the fish swimming from these scans, which is really cool. Okay, so yeah, I'll leave it there. So this is actually some heart data, which may or may not play. It's not going to. But yeah, just to thank everyone involved in the imaging. So obviously, it's not just me involved in all these studies. So yeah, all these people are involved. So we have lots of different users that come and use the center. And I've tried to include a nice kind of snapshot of a lot of different things we do. And yeah, I'll leave it there, and I'm happy to answer any questions if you have any.
Thank you very much, Jack. It was a great talk, and actually really, really interesting data, especially with the last one with the bats as well. So yeah, audience, feel free to send me questions, and I would address them. There are actually some questions already popped up. One of them is they have asked for your macrophage injection because you are studying the the kidney injury. Why did they inject them into the heart and not into the sort of renal capsule?
Okay, because as I kind of touched on later on, we don't think you necessarily need, especially with the macrophages, to administer them to the organ itself. And I think you're more likely to cause more damage to the organ if you try and do this. There is studies that do that, definitely. I've seen them. But we have definitely shown that macrophages can migrate after injection to the heart to the kidney itself. So we don't think it's necessary. It might be that that would work as well, but we don't think it's necessary with macrophages.
Okay, cool, thanks. Actually, I had a question following that study as well, regarding that you clearly showed that your macrophages do migrate to the site of injury. But in terms of the recovery or regenerative sort of data, it didn't show that much of an effect. Why do you think that was the case?
No. Yeah, so yeah, it definitely does not show much of a case. When I was signing that study, I was really trying to develop the imaging methods to do this rather than actually do an efficacy study. So I did see a hint of efficacy. It was promising, but I think the problem is the model I was using is a really severe model of injury, and I don't think much would have rescued these mice. I think they were actually quite sick at this point. I think if we used a more subtle injury model, we might see that. And I'm actually writing a fellowship grant now to try and do that. That we might see a better effect with the therapy. But yeah, you're correct. The therapeutic benefit wasn't huge in that study.
Cool, thank you very much. There's some questions that came up regarding the center, the amazing center that you have with all these various different imaging modalities. So when did you actually started, and do you actually give services just for the sort of university or outside as well?
So the center was opened in 2015, but the grant, in order to get the center, I imagine that's before I was here. So I imagine at least a couple of years before they were trying to establish the center itself. Once they got the grants, they hired Harish, who became the academic lead, because they really didn't have anyone who had any expertise in MRI, so they needed to get someone. And then once he came, the center itself opened, and we moved all the modalities here, because we used to actually have them spread out in the university's animal center, but now we moved them all into one roof. and since then we've written more grants to receive the PET, SPECT and CT, which we only started a couple of years ago. And the second part of the question, our user base is
do you actually
mainly the
provide
university. Sorry, say again.
Sorry, no, I thought you forgot my second part of the question. I was just repeating it.
No, sorry, no. So it's mainly the university, but we do encourage outside users. We don't have a huge amount of them. The problem is that the laws that govern animal research are very strict. So getting a license to be able to transfer animals from one center to another or one university to another is very difficult. And if I'm being totally honest, our animal team here at the university do not make it easy for us. So we try and accommodate people, but it's extremely difficult and it puts people off, to be honest. You want more business, we need more business, but it's difficult to get that.
Yeah. And do you have a count of number of projects that you've done so far in this last 5 years-ish?
Off the top of my head, I don't think I could tell you that. I might be able to dig that out and tell you by email, but I couldn't answer that. Harish would probably be able to answer that, but I'm afraid I don't know off the top of my head. But we do have a number of different groups. We have a number of groups in pharmacology that use the systems. We have groups in cancer medicine that use the systems. Like I was saying, we have anatomy groups, we have musculoskeletal biology groups. So yeah, we do have a lot of people, and the user base is increasing. We have a number of publications. I think on our website we'd hopefully list them. We should have done. If you want to check.
Cool. Thank you very much. Just one last question. Personally, I have how much are the costs actually for running a scan? So let's say I don't know, a PET scan. How much that would be?
Okay, so the PET scan, I think off the top of my head, is £180 an hour. And I think that's similar for the MRI too. And we say to users, you can book a minimum of half an hour, so 90 English pounds for half an hour. The IVIS system is our cheapest, that's £80 an hour. And that's the same for the MSOT as well. So in comparison to other centres in the UK, we're actually quite cheap, I think, quite reasonable. So I think in London, the prices are much more expensive, as you might expect. But we try and keep the prices usable, because that's another one of the main limiting factors. People want to use the system, but they need the money from grants, and it's hard to acquire money. So we try and keep it as affordable as possible.
Cool. Thank you very much, Jack. And with that, I would like to end, because I know you have to go and pick up your kids as well. And on that note, actually, I want to congratulate all the audience, because in Iran today is the Women's Day and Mother's Day as well. So I congratulate all of the ladies of the audience as well. And with that, I would really, really sincerely thank you, Jack. And it was an amazing talk, and really looking forward to have a talk from your center on our sort of annual symposiums that we have. And this year would be our 4th annual symposium.
Okay.
So TPCF is just like your center. We are a sort of preclinical core facility, and we're quite new, not as old as you guys. So roughly around 2.5 years. And so we provide mostly studies just like yourself to university students, PhD students, and postdocs whoever's interested in preclinical imaging. And also we're trying to educate the whole community, whether in Iran or even outside. Therefore, we have all these TPCF webinars that we are very honored that you actually accepted and gave this wonderful presentation. And with that, I would like to end the session. And once again, thank you very, very much, Jack, for your amazing talk. Thank you very much.
Thank you very much.
Thank you. And just to finish there, I would actually ask the admin to actually advertise the next month's sort of TPCF webinar. So next month, we are going to have a sort of a TPCF webinar from one of the scientists, actually, he was based in America, as far as I remember. And we really look forward to everyone to have you for that session as well. And with that, I would like to end the session, and thank you for attending this webinar. And once again, sincere thanks to Dr. Sharkey for his amazing talk about Liverpool Preclinical Imaging Center. So our next month's talk would be on magnetic resonance histology by Professor Alan Johnson. And so, yeah, looking forward to that as well, to see you all over there. And with that, good night to everyone, or good evening, and hopefully see you next month. Thank you very much.